Genetic Drift and You, featuring Mr. Verdi and Aaron Mole. Okay, so genetic drift. Here's what it is. It it is essentially this random drift, right? When we mean drift, we mean like you can think Moana. Moana's boat got in that ocean. It kind of drifted over to Maui's island, right? She didn't steer it, right? Well, what we want you to see is that there's two ways where genetic drift happens. First off, okay, but it's another mode of evolution. All right, natural selection is the only thing that causes species to evolve to match their environment. Okay, so the reason uh, we're so good at talking, right? The reason our brains are so big is natural selection, right? That was a selective pressure that made it important for humans to be able to keep track of relationships and communicate. All right, natural selection led us to that. Natural selection is the reason, bugs are camouflaged. That's natural selection, right? Sexual selection, like we talked about the last time we were together, that's why animals are pretty as they try to attract mates. Genetic drift is a weird one, right? It is a fluctuation as we look down here. It's a fluctuation due to chance, not necessarily more fit individuals surviving and reproducing. Okay, so if we look at here, when Bambi meets Godzilla, it's a classic in American film. Here's Bambi, and she was a good, tough deer, I think. I don't really remember all that happens with Bambi. I think she might have died anyways. But she might be the biggest, strongest, fastest deer. If a random event happens and Godzilla stomps on her, there's, notice her legs. This is pretty graphic. Sorry, kids. Okay, no deers were hurt in the making of this cartoon. But if, let's see here, if she's bigger, stronger, faster than all the other deer, but something falls on her head, it really doesn't matter how good her genes were. And that's what genetic drift is all about. Okay, this random change in a population that doesn't select for the more fit or the most adapted individuals. Okay, it really is chance events altering the genetic frequency. Okay, so here's the two ones we have to know and be able to differentiate between. Okay, the founder effect and the bottleneck effect. In the founder effect, we would have an original population and some random individuals choose to leave it and find a new island, imagine, right? Or separate from the rest of their general gene pool. Well, what we want you to recognize is in this new island, there is 6 blues to 1 yellow marble, right? We're just using marbles. Okay. That was not the original ratio, right, of the original population. But because of a random sampling of individuals that got put in this new separate island, this population will have a different gene frequency and then be a little different than the one it came from. Okay, and we'll use actual examples to show you. So here's another example of genetic drift. You've got, imagine you've got all these different marble colors, and there's this bottleneck effect. Effectively, a lot of individuals die. Think a hurricane, right? A hurricane is going to kill a lot of chipmunks, right? Whether or not you're camouflaged and faster and able to hide from hawks and owls, right? And then just a random group survives. That's what the bottleneck event shows. Notice they're very similar. We get a new ratio of marbles. This represents phenotypes and genes in both. That's not necessarily reflective of the original population. And in fact, maybe the purple ones were the most fit. They blended in the most, let's say, in this bottle, but none of them survive a hurricane. And then this new population is stuck without those genes. Okay, so here's just some examples. The founder effect, the old order Amish of Pennsylvania. It's the founder effect. They didn't necessarily find a new island, but they separate from the original population. And there's this gene where a lot of Amish in this particular population get 6 fingers. Okay, and they came from an original population, but the gene pool was so small that left that this gene sticks around. This 6 gene is called polydactyly, right? So that's an example of founder effect. This happens in a much higher ratio in this population than the original population it came from. Again, random individuals left the current population to start their new one, and there's a different ratio of genes, okay, that might not necessarily be more fit. Okay, it happened in panthers. This is the bottleneck effect. Okay, panthers. Well, there's this original diversity in genes in this panther population. Well, by the 1970s, there was about 20 to 30 Panthers left in Florida. Okay. Now, even though, and so what do you get? These little two marbles here, right? The surviving population rebounded, and Panthers are doing better in Florida. But they have this very, very low genetic diversity because they all got so many died as they were hunted in Florida in the 1970s. Okay, so they're basically inbreds, right? They're mating with each other's third cousins. Okay, kind of weird. You might expect something like that in West Geauga, but not with the Florida Panthers. Okay, sorry, West G kids. Okay, but that's for you guys. All right, so we have a listener. Last one, I'll let you guys go. Okay, you can see my countdown here. All right, the greater prairie chicken. Well, pre-bottleneck effect, Illinois 1820, when these things roamed free, okay, they were everywhere. Well, they were effectively selected against pretty well for the next 173 years, and now the chickens are down to like these little populations. Okay, so what do we have here? You've got the population size and the number of alleles per trait. Okay, how many different versions of genes they have? Well, now it's 5.2. Sorry. Oh, here it is. Sorry. I'm sorry. Here's how you're going to read this. In 1993, it's down to 3.7 compared to 5.2. Well, what effect does this have? 93% of eggs are hatched from 1930s to 1960s. Now the eggs that are hatched, only about 50% hatch. I mean, these things are having birth defects and they have issues generating eggs and being born because there's such a low genetic diversity, right? Again, it's kind of like if we kill everybody else eventually, and these guys are going to rebound, you're going to be reproducing with your cousins. Okay, and that's not good for individual for the offspring. Kansas, there's no bottleneck effect. So here's the experiment, right? No bottleneck in 1998. The number of alleles per locus stays high. It didn't drop. 99% of eggs are hatched. Okay, so that's genetic drift with two examples of bottleneck and founder effect. Thanks for coming, guys. There's more to go here, but I've got to run. So see you, kids. We'll talk soon. Mole.
Hey, Birdie.
Did you like it?